A queen bee is not born. She is manufactured, and the most disturbing part is not how it happens. It is who decides, not a king, not a god, not even the queen herself. The colony decides. 50,000 insects with no central brain between them collectively choose which larva becomes royalty, which ones remain sterile workers for life, and which rivals never get to emerge from their cells at all. They make this decision chemically, silently, and without a single mistake. They have been doing it for over 100 million years, and the mechanism they use is so precise, so ruthless, and so biologically extraordinary that it forced scientists to completely rethink what identity even means at the cellular level. Here is the central mystery. Every single bee in a colony, the workers who forage for pollen, the workers who fan the hive, the workers who guard the entrance, and the queen who lays up to 2,000 eggs per day. Every single one of them begins as a genetically identical egg. They share the same DNA. They come from the same mother. Under a microscope, a queen egg and a worker egg are indistinguishable, and yet the adult organisms that emerge from these identical starting points are so different that for centuries, naturalists refused to believe they were even the same species. The queen can be 40 to 60% larger than a worker. Her abdomen is elongated, built to hold ovaries that contain hundreds of thousands of eggs. She can live for 3 to 5 years, sometimes even longer, while a summer worker bee survives for only about 6 weeks. The queen never forages. She has no pollen baskets on her legs. Her sting is smooth, allowing her to sting repeatedly without dying, unlike a worker whose barbed stinger tears out of her body in a single defensive act. She is in almost every biological sense a different animal, and she was made that way, deliberately, by the colony itself. The mechanism behind this transformation is one of the most fascinating discoveries in all of modern biology, and scientists are still unraveling its full complexity. For most of the 20th century, the explanation seemed relatively simple. Worker larvae receive a diet of worker jelly, a secretion produced by nurse bees that is nutritious but limited. Queen larvae, on the other hand, receive royal jelly, and they receive it in enormous, almost extravagant quantities. Their cells are flooded with it. While a worker larva is fed just enough to develop and survive, a queen larva swims in royal jelly throughout her entire larval development. The royal bath never stops. And that difference in feeding, scientists believed, was the switch that unlocked the queen developmental pathway. The logic seemed clean, almost satisfying. One substance, one fate. But in the 2000s and 2010 cells, research began to complicate this picture in deeply interesting ways. Scientists started to realize that the story was not simply about what the queen receives, but also about what the worker does not receive. A compound called p-coumaric acid, found abundantly in pollen and therefore present in worker jelly, turns out to act as a powerful suppressor of queen development. When larvae are exposed to p-coumaric acid, it activates certain genetic pathways that push development toward the worker body plan. Remove that suppressor, flood the larva with royal jelly instead, and an entirely different set of genes switches on. The queen pathway unfolds. What this means, and what makes it genuinely astonishing, is that the worker bee body plan may in fact be the suppressed version of the queen. The queen is, in a very real sense, the default. The worker is the modification. The colony, through the act of feeding, is constantly pressing down on the development of its own members, keeping them sterile, keeping them small, keeping them locked in a role they were chemically prevented from transcending. If you are reading this and feeling slightly unsettled by that idea, you are not alone. The implications are strange. The colony is not simply nurturing its workers, it is at a biochemical level suppressing them, constraining them, deciding their futures before they have any say in the matter. And it does this not with malice. Bees have no malice, but with the cold, elegant precision of evolutionary optimization that has been refined over millions of years. Now, if you are finding this as mind-bending as we do, take a second to hit that subscribe button, because we are just getting started, and what comes next is even more unsettling. The actual making of a queen begins in one of two ways, and both of them involve a kind of controlled crisis. The first is called supersedure. This happens when a colony's existing queen is aging, injured, or producing fewer eggs than the colony needs. The workers detect this decline through pheromones. A healthy queen produces a complex cocktail of chemical signals, including a compound called queen mandibular pheromone, or QMP, which suppresses the ovarian development of every worker in the hive, and signals that the colony is stable. As the queen ages, her QMP production drops. Workers begin to sense the change. And then, with no announcement and no deliberation, a decision is made. Queen cells are constructed. The second scenario is called swarming, and it is perhaps the more dramatic of the two. When a colony grows large and prosperous, when the hive is crowded and resources are abundant, the colony enters a reproductive phase. It prepares to divide itself. The old queen will leave with roughly half the workers to found a new colony elsewhere. But before she goes, the remaining bees must have a new queen waiting to emerge. And so again, queen cells are built. These queen cells are architectural masterpieces in miniature. Unlike the standard hexagonal cells of a comb, queen cells hang vertically from the comb's surface, oriented downward, shaped like elongated peanuts, roughly the size of a human thumb. They are reinforced and thickened. They are built for something important. Inside each one, a larva no more than a few days old floats in a reservoir of royal jelly, consuming more than she can possibly need, her body beginning the extraordinary process of becoming something unprecedented. The timeline is tight and unforgiving. From the moment an egg is laid, it takes about 16 days for a queen to emerge, compared to 21 days for a worker. The queen develops faster. She is pushed through development at an accelerated pace, her body reorganizing at a cellular level, her ovaries multiplying and differentiating in ways that no worker's body ever will. When she finally chews through the wax cap of her cell and emerges, she is soft, pale, and already formidable. And here is where the terror enters the story. Because in most cases, she is not the only queen. The colony has built multiple queen cells, a hedge against failure, an insurance policy written in wax. And the first queen to emerge faces an immediate biological imperative that is not nurturing, not communal, not gentle. It is assassination. The newly emerged queen moves through the comb with purpose, locating every other queen cell she can find. She tears into each one. She stings the developing larvae inside, killing them before they can emerge. If another queen has already emerged, the two will fight, and they will fight to the death. No quarter is given. No compromise is possible. There can only be one. Scientists have observed that in some cases, worker bees will actually guard developing queen cells from a virgin queen who is moving to destroy them, particularly during swarm preparations, when a backup is still needed. The workers place their own bodies between the murderous queen and the cells she is trying to destroy, holding her back temporarily, managing the succession like a council of advisors controlling an unstable monarch. It is by any reasonable measure, a breathtaking example of collective intelligence operating without any central authority. If you are watching this and your mind is already racing with questions, drop them in the comments below. And if you haven't already, hit like. It genuinely helps this content reach more people who would find this as extraordinary as you do. Once a queen has eliminated her rivals, her next challenge is mating, and this too is unlike anything in the rest of the insect world. The virgin queen leaves the hive on what are called mating flights. She will fly to a drone congregation area, a location where male bees from dozens of different colonies have gathered, sometimes thousands of them, in a floating buzzing cloud that can persist in the same geographic location for years. The queen flies through this cloud and mates with anywhere from 10 to 40 individual drones in a single flight, or across multiple flights over the course of a few days. Each mating is brief and violent. The drone's reproductive organs are literally torn from his body during the act, and he dies shortly afterward. The queen collects and stores enough sperm in a specialized organ called the spermatheca to fertilize eggs for the rest of her life, which can span years and represent millions of individual offspring. She will never mate again. The genetic diversity captured in a single mating flight is extraordinary, and it turns out, critically important. Because she has mated with so many different males, the workers she produces are not sisters in the simple sense, they are half-sisters, daughters of the same mother but different fathers. Research has shown that colonies with higher genetic diversity among their workers are more resistant to disease, better at regulating temperature, more behaviorally flexible, and more efficient at foraging. The queen's profligate mating strategy is not recklessness. It is a sophisticated biological mechanism for ensuring colony resilience. The chaos of that mating cloud produces a hive that is more adaptable, more robust, more capable of surviving the unpredictable challenges of the natural world. There is one more dimension to this story that deserves attention, because it sits at the edge of what science currently knows. Royal jelly is a white gelatinous secretion produced by glands in the heads of young nurse bees. It contains proteins, fatty acids, sugars, vitamins, and a family of proteins called royal actins that are, as far as we know, unique to bees. The royal actin proteins appear to activate growth pathways, triggering the accelerated development and enlarged body size characteristic of queens. When researchers removed royal actin from royal jelly in laboratory conditions, larvae fed the depleted jelly, developed more like workers even when other nutritional factors were controlled. This suggests that royal actin is not merely nutritious, it is a signal. A molecular message that says in the chemical language of biology becomes something extraordinary. But here is where the speculation begins, and we want to be clear that what follows is the frontier, not settled science. Some researchers have proposed that royal jelly may work partly through epigenetic mechanisms, changes not to the DNA itself, but to the way the DNA is read and expressed. Chemical tags called methyl groups can be added to or removed from DNA, switching genes on or off without changing the underlying sequence. Early studies in bees suggested that methylation patterns in queen and worker larvae diverge dramatically during the larval period when feeding differences are most pronounced. If confirmed, it would mean the colony is not just feeding its future queen, it is reprogramming her genome's expression in real time, writing a new biological identity into the very architecture of her cells. The research is ongoing, the picture is still incomplete, and scientists are careful not to overclaim. But the direction the evidence points is remarkable. There is something almost philosophical about all of this. The queen did not choose to be queen. The workers did not choose to be workers. The drones did not choose to die in the act of reproduction. Every role in the hive was assigned through a process so ancient and so automatic that the word choice barely applies, and yet the system works. It has worked for 100 million years through ice ages and extinctions, through every catastrophe the planet has thrown at life. The hive does not need a leader to function. Everything emerges from the collective behavior of tens of thousands of individuals, each responding to local information, each playing a role assigned by chemistry before they ever took their first flight. Right now, in a meadow somewhere, in a forest hollow, in the wall of an old building, this process is unfolding. Eggs are being laid. Larvae are being fed. Queen cells are being constructed in secret, hidden in the lower margins of the comb, invisible to any observer who does not know to look. And somewhere in one of those cells, a tiny larvae is floating in a bath of white jelly, her genes beginning to respond, her body beginning its extraordinary transformation into something that will outlive every other bee in the hive. Something that began as identical to every worker around her and became utterly unlike any of them. Not because of fate, not because of destiny, because of what she was fed. The next time you hold a jar of honey, or walk past a flowering tree surrounded by the sound of bees, consider what made that possible. Consider the queens that were made, and the queens that were killed. Consider the workers whose potential was chemically suppressed in service of something larger than themselves. Consider that one of the most complex and successful societies in the history of life on Earth runs entirely on chemistry, instinct, and a kind of collective wisdom that has no name and no single mind behind it. That is not magic. That is biology. And in some ways that is more astonishing than magic has ever been. If this left you seeing the world differently, share this video, leave a comment and subscribe, because the world is stranger and more beautiful than most of us are ever told. We will see you in the next one.